Well, thank you, Peter. Hi, my name is David Stamler, and I am the CEO of Alterity Therapeutics, and I'm very happy to tell you about our programs today. These are our forward-looking statements, which I encourage you to review, and our regulatory documents. The name Alterity means the state of being different, and we are dedicated to creating an alternate future for patients living with neurodegenerative diseases and to really move them to a healthier, different state. We aim to do this by addressing the underlying pathology of the disease with our molecules. We have this very skilled R&D team that has 3 FDA approvals under our belts, and we are advancing our lead molecule ATH 434 for a rare disease called multiple system atrophy, which has no approved therapies. We have our ongoing phase 2 trial, which is a randomized double-blind placebo-controlled investigation in our lead indication, as well as a strong patent portfolio. I've had the fortune of leading 3 FDA approvals in the neurology area over the last several years. Most recently, we had 2 drug approvals in 2017 at Auspex Pharmaceuticals. We were acquired by Teva in 2015 for over $3 billion. At that time at Auspex Teva, Margaret Bradbury and Cynthia Wong joined me in that endeavor, and they have since joined Alterity and are part of our strong team. Today we'll be talking about Parkinsonian disorders. And this represents a significant unmet need. Parkinsonism, which is a motor syndrome that you've probably seen in friends or family members, is the slowed or stiff movement that is a major source of disability. But in addition to Parkinson's disease, there are other disorders, Parkinsonian disorders, that also have this motor symptom. These are called atypical forms. And they're called atypical because patients have other symptoms that cause as much or more problems for these patients. And they also don't typically respond to therapies that work in Parkinson's patients. Importantly, the available therapies address the symptoms, but do not address the underlying pathology of the disease. This is our lead molecule, as I've told you about, with our ongoing phase 2 study that we aim to expand shortly. We also have an ongoing natural history study in the same patient population that represents a longstanding collaboration with Vanderbilt University, and I'll tell you more about this later. And finally, we have a very active drug discovery program with recent composition of matter patents in the last year or so, and we aim to bring new IND candidates forward in the near future to treat major neurodegenerative diseases. So our strategy really is centered on targeting this protein called alpha-synuclein. And this is a normal protein, as you see on the left side of the slide, that is important for neurons to communicate with one another. However, in disease, this protein actually misfolds and it forms clumps. And you can see that on the right side of the slide. So in the physiologic state, it's here, but in disease, it forms these fibrils. And that causes problems because when it does that, the nerve cells don't work well. And also the body tries to get rid of the clumps of protein, which causes more problems downstream. So we really aim to prevent this protein misfolding by binding and redistributing excess iron in the brains of patients. And we and others have shown that that iron actually causes the protein to misfold. So our strategy really is to bind and redistribute the excess iron in the brains of these patients to reduce the alpha-synuclein misfolding and aggregation, and also block oxidative stress at the same time. And in doing so, we aim to rescue neurons and preserve function in these patients. These are data from about 30 years ago. These are post-mortem data, so pathology data from brains of Parkinson's patients. These investigators showed that in the areas of pathology, these patients have elevated brain iron compared to controls. Now, they also showed the same thing in patients with multiple system atrophy, but in multiple other brain regions. Now, if you fast forward about 25 years, investigators have now developed advanced MRI methods to show that we can quantify the iron in the brains of these patients. And you actually see in this picture at the tip of the orange arrow, this dark red staining material corresponds to increased iron in the brains of MSA patients. And that corresponds to the same where these orange arrows are pointing in here. So this basically shows that we can image the iron in a living patient, and thereby we can track the activity of our drug in a living patient. So regarding clinical development, 434 is shown on the left side of the slide. It is a small molecule that we have shown crosses something called the blood-brain barrier, which is important so the drug can actually reach the site of action and interact with the proteins in the brain. The drug actually has received orphan drug designation both in the US as well as in the European Union. And finally, 434 is an oral drug, which is important for ease of use and is preferred by clinicians and patients. A few words about multiple system atrophy. As I mentioned, it's a Parkinsonian disorder, so they have motor symptoms, as you can see in the second panel. But the patients also have significant involvement of the autonomic nervous system. So they have difficulty maintaining their blood pressure, bowel, and bladder function. Unfortunately, the disease is rapidly progressive, and more than 50% of patients become wheelchair bound within 5 years of symptom onset. A typical life expectancy is about 7.5 years after symptom onset. So there really is an urgent need to develop new therapies for these patients. Before conducting a phase 2 study, we've done a phase 1 study in healthy volunteers, as you can see on the slide, a very well conducted or well designed study, well controlled, looking at single doses up to 600 and repeated doses up to 250 milligrams twice a day, including older adults. And we saw very strong and reproducible pharmacokinetics in that patient. And we also showed that we could deliver the drug at good concentrations into the spinal fluid, higher than we could achieve or that were associated with efficacy in animal models of disease. The drug was also well tolerated, with no adverse events leading to withdrawal, no serious adverse events, and similar rates of adverse events between active treated patients and placebo treated patients. And all adverse events were mild to moderate in severity. This is the phase 2 study design I told you about. This is a robust design. It is a randomized, double-blind, placebo-controlled study that is examining the efficacy and the safety of our lead molecule, ATH 434, in patients with early MSA. We're looking at approximately 60 patients in 30 sites across the globe, including Australia and New Zealand. And patients will be randomized to 12 months treatment with either of 2 dose levels of 434 or placebo. And our primary endpoint is that change in iron on brain MRI that I showed you on that prior slide. We're also looking, importantly, we're looking at clinical endpoints that look at activities of daily living and motor performance. These are important endpoints to support the ultimate approval of the drug. Now, why are we looking at brain iron? This is, we and others have shown that if you actually compare the brain iron with disease severity on this x-axis, and disease severity is measured by this widely used scale called the Unified MSA Rating Scale, we and others have shown that the amount of brain iron correlates with disease severity. We are also developing additional methods to try and improve the precision of how we measure this brain iron in the study. From a commercial standpoint, you can imagine with such a severe disease and with no approved therapies that address the underlying disease, there is a significant opportunity. This assessment is based on interviews with 30 US neurologists, half specialists, half generalists. And those clinicians recognized that there was a substantial unmet need, and they had a very strong intent to prescribe because the drug does address the underlying pathology of disease. They also appreciated the ease of use with an orally administered drug. So based on all these, this feedback, as well as rather modest assumptions regarding the commercialization of the product, we estimated peak sales of up to $725 million US, and that's US dollars in the United States alone. So in summary, we are targeting an orphan disease that has no approved therapy. We have a development team with a proven track record and multiple FDA approvals. We have launched our phase 2 study, and we've got a very strong discovery team that is identifying new molecules to take forward into major neurodegenerative diseases. We've reached these first 2 milestones on the right side of the slide, and we expect to be launching the trial in the second half of the year in Europe, as well as in the United States. Thank you.